Hello everyone, welcome to A-Level Biology with Dr. Demi. I am Dr. Demi, as you already know, and if you're watching this video, I want to say welcome to memberships and thank you so much for signing up for a membership. Always remember you can cancel at any time, but I promise you this will be totally worth your while. I will be doing solutions to some of the specimen papers, to all of the specimen papers as a matter of fact, for the upcoming 2022 exams, and I'll explain what a specimen paper is shortly, but I'll also be taking you through some tips on how to succeed in the exams, how to prepare, as well as do extra past papers with you so that you can understand. And I plan to do a couple of live streams during the month of April, which will be for members only, so it'll be dependent on people signing up for memberships. Even if they're only 2 members, I'll still organize a live stream and we will have a class together where we just discuss anything that is not clear to you throughout the syllabus for biology. Now, a specimen paper is a paper that sort of introduces students to what they should expect in the exam after the syllabus has changed. So if you look through the playlists that I have on the channel, you will remember that I did a video about the syllabus changes, and so this paper is basically an introduction to what you should expect. I'm going to do paper 1, 2, 4 and 5, and I'll also try to look at instructions for paper 3 just to give you an idea of what you can expect as well. And there is no paper 3 anywhere else on the channel, so this is going to be highly exclusive for those who have signed up for memberships. So yeah, let us get right into it, and I hope that you have a lot of fun. So just want to put out a disclaimer that it is the end of my day here in the UK and I am quite tired, but I promised that this video would be ready before tomorrow, which is why I am recording it now. So please forgive if my voice sounds a little bit down or a bit exhausted, but I hope that I can keep you engaged long enough to answer these questions. All right, let's go for it. For question number 1, it says, which definition of the magnification of a drawing of a leaf is correct? So when I say a question like this, the first thing I say to students is that you need to first of all get what magnification is. What is magnification? According to biology, it is image size over actual size. All right, and here they say that what is the definition of magnification of a drawing of a leaf? Okay, and so if you're dealing with a drawing, that means that the drawing itself is the image. All right, that's what you call the image in that case. And if you have the leaf in your hand, that would be the actual size of the leaf. So in this case, let's look at the options we've got. The actual size of the leaf multiplied by the magnification. That's obviously wrong because you're trying to define magnification, and we know this is magnification here. The measured difference in size between the leaf and the drawing of the leaf, that's not true. The increase in size of the leaf when observed using a microscope, again, that is not true. The size of the drawing of the leaf divided by the actual size of the leaf. So D is the correct answer here. And if you have any questions about why it is not, for example, B or C, please post a question in the comments, and I will respond to you. As a member, I am obligated to respond, to treat your comments as priority. So please post them in there and I will respond as soon as possible, or I'll respond really quickly, to be honest. Okay, let's look at number 2. The electron micrograph shows part of 2 cells. Which labeled features identify these cells as eukaryotes? So let us look at this. So over here we can see, I'm just going to go XWY for whatever reason, or we can actually start with W. So W over here seems to look like the chloroplast when it has been stained. X is pointing to what I believe is the cell wall, and Y over there is pointing to the cell membrane. So which feature identifies these cells as eukaryotic cells? Now, something that you need to remember is that prokaryotes also have a cell wall that is made up of peptidoglycan. So you can't use the cell wall as a distinctive feature with which you will judge whether or not this is a eukaryotic cell. So that sort of knocks X out of the picture. Well, X is X in that case. And if you look at Y seems to be pointing at the cell membrane. but there are other organisms that have cell membranes as well. Again, some of them are simply prokaryotes. So Y is not enough of an indication to let you know that this is a eukaryotic cell. The only thing that points at it possibly being eukaryotic is the stained picture, the stained image of what looks like the chloroplast granule granular arrangement. And so in this case, I would say C is the correct answer. All right. Okay, let's look at question 3. It says plant cells are stained and viewed using a student microscope. The light source is natural light, what could clearly be visible at 400 times magnification. So in this case, when you look here, you can see it has the cristae of the mitochondria, the grana of the chloroplast, the nucleoli, and the ribosomes. I did a shorts video where I explained to you all that you can't see some of these things under the light microscope. Even when you stain them sometimes, they're probably not as visible as they should be. And I'll explain why to you. The reason why this question is telling you the magnification and also telling you that it's a student microscope with this kind of light source is because the question is basically telling you that with a light microscope that has a magnification of 400 times but poor resolution, because remember with the light microscope you get magnification at the expense of resolution. That means you can make things bigger, but you can't necessarily make them clearer. So that means that you will not be able to see things that require you to have clarity, such as the cristae of the mitochondria. The cristae of the mitochondria is the inner membrane of the mitochondria, so that will not be visible. The grana of the chloroplast will also not be visible, but what you will be able to see is definitely the nucleoli, and I'll tell you why. When you stain a plant cell or even an animal cell, the chromatin that is held inside the nucleus or inside the nucleoli, which is the form of DNA that is tightly unwound, that is tightly wound, but not in the form of chromosomes, will hold on to the dye quite well, and for that reason you will be able to see the nucleoli. Chances are you will not be able to see the ribosomes, and that's possibly because they won't stain well because they are so tiny, but you certainly will be able to see the nucleoli, so the answer here would be C. Now let's look at the next question. Which lengths are equivalent to 1 micrometers? This is usually one of my favorite questions because this is what we basically have to teach students right at the beginning of the course. And the way I just teach it to them is I tell students to remember to write down the different steps to getting the different units. Okay, so you know that if you're going that way, you're multiplying by 1000, right? So that means that 1 meter is going to be equal to 3 zeros, so it'll be about 1 billion nanometers. Okay, because you just put all the zeros together for those who might not be able to solve this. So what then is the equivalent of 1 micrometer? If you're looking at 1 micrometer, you want to see what it's equivalent to. So for nanometers, first of all, you can say 1 micrometer is simply equal to 1000 nanometers, right? So this is your easiest one. So you can come here and say, do you have anything that's 1000 nanometers? No, you don't. The nanometer that you have is 0.001, so that's wrong. You know that it is certainly not number 2. If you look at number 1 micrometer, if you're going back to millimeters, you have to divide. So that means it's 1 divided by 1000 for you to get it in millimeters. And that is going to be 0.001 millimeters. If you look here, this is not the answer. It's not 1000 millimeters. Look at anything else that's millimeters. That's also not the answer. But you do have this one here, which seems accurate. So that's correct right over there. Okay. And let's see, for nanometers, we did say that 1 micrometer is equal to 1000 nanometers. This one is also correct. You're only required to know 2. You definitely know it can't be 1 million. So that's wrong, and so you're left with 3 and 6, and that leaves you with the answer D. I hope that was easy to follow for everyone. If you did not get it, please post a question in the chat and I will be able to respond to you. Okay, let us look at question number 5. It says here, some secretory cells synthesize and release glycoproteins. What is the correct sequence of some of the events that occur in a secretory cell? So it says some of the events. Well, first things first, based on what this question is asking, is that you will need to know protein synthesis in a little bit of detail, I mean just the basics, as well as know bulk transport, and also sort of have an idea of what organelles in your cell are involved in the protein synthesis process. But let's look at what we've got. The first one says exocytosis. Now that would most likely be your last option. That would most likely be the last step, because exocytosis means the release of compounds or proteins or organelles or whatever it is outside of the cell. Endocytosis means that it is engulfing into the cell. So that would be the last thing. It says products accumulate in secretory vesicles. In this case, I would put that as number 3 because the vesicles are the ones that are released via exocytosis. mRNA binds to ribosomes and then synthesis of glycoproteins. If you remember the protein synthesis sequence, the mRNA would have to bind to the ribosomes first before you can make the glycoproteins. And of course, if you are unsure of this, please, I would encourage you to watch my protein synthesis videos so that you understand it. So in this case, the answer would be 3, 4, 2, and 1. So let's see 3, 4, 2, and 1, and that is B over there. So very easy and straightforward. Now let's look at number 6. Number 6 here says a scientist used a blender to break open a sample of animal cells and release the organelles. The extracted mixture was spun in a centrifuge at low speed. The heaviest type of organelle sank to the bottom of the centrifuge tube to form a solid pellet number 1. The liquid above pellet number 1 was poured into a clean centrifuge tube and spun at a higher speed, and the next heaviest organelle sank to the bottom and formed pellet number 2. And that was repeated again to obtain pellets number 3 and 4, each of which contained a single type of organelle. What is the function of the organelle extracted in pellet number 3? Now this question can be very confusing for students, and that's particularly because cell fractionation might be something that you have not studied or done in class, and I might have to do a video on this considering that it is also coming in 2022, or you should expect it in 2022. It doesn't mean it will be there. But what this question is basically asking you is to think of the sizes and the mass of the different organelles of the cell in the cell, and consider which ones would be the heaviest and then which one would be the second heaviest, and so on and so forth. Now in this case, you can think of the fact that if you're synthesizing proteins, you're going to be dealing with ribosomes. So you know that ribosomes are not heavy because they're really, really small. Okay, so these are the ribosomes. The one that's responsible for the synthesis of mRNA is actually a protein called RNA polymerase, or rather an enzyme called RNA polymerase. Okay, so that might not be your but oh, I might be wrong here because it's asking for an organelle. Then you might not be thinking of RNA polymerase synthesis of mRNA. Then you would be thinking of maybe the nucleus, because that's where the mRNA would be made. Sorry about that. So you go with the nucleus here. The production of ATP by respiration would be the mitochondria, and then digestion of old cell organelles, that would be the lysosomes. So those are the functions of those different organelles. Now these are more or less the 4 different pellets that you have extracted. So because these are the only options they've given you, you should assume that these are the 4 different pellets, which means that the heaviest of these would be the nucleus. Okay, so that means the very first pellet was the nucleus, and the reason why the nucleus would be so heavy is because it contains all of the DNA that's held in the cell. The mitochondria would certainly be number 2, and obviously you know the ribosomes are not bigger than the lysosomes. The lysosomes are bigger, so this would be number 3. And so when they say what is the function of the type of organelle extracted in pellet 3, it would be A, which is digestion of old cell organelles. I hope that that was clear. Again, if it wasn't, just post a comment and I will be able to answer your questions. Okay, question number 7. What is the general formula for amylose? Now, this is a question many students might be tricked on because they often just think, well, amylose is a polysaccharide. It is made from repeating glucose units. So I just need to pick the formula for glucose and then put an N next to it. That is not correct. Always remember that when you have a disaccharide or a polysaccharide forming, you have the release of water. That is why it is called a condensation reaction. And so for that reason, it means that you lose 2 hydrogens and you lose 1 oxygen, right, in each joining of these different bonds. So the formula for amylose in that case would then be the release. You will have to deduct H2O from whatever this is in the bracket. So in this case, that would be 2 hydrogens deducted from here would make it C6H10, and 1 oxygen deducted would make it O5, and that's why the answer here is D and not C. All right, in case you were thinking it's C. Let's look at question 8. Question 8 says, which statement describes how the molecular structure of starch is suited to its function? Amylose has a branched structure and amylopectin is coiled to give a compact molecule for transport. That's wrong. Amylose is actually a straight chain and amylopectin is the one that has the branches, so that can't be correct. The second one says, in the breakdown of amylose and amylopectin, many condensation reactions release stored energy. This again is wrong because when you're breaking down amylose and amylopectin, you are adding water, and that is not a condensation reaction. That is a hydrolysis reaction. The release of water is condensation. The addition of water to break the bonds is hydrolysis. Let's look at C. In the formation of amylose and amylopectin, many hydrolysis reactions allow the release of stored energy, so that is a possibility. But let's look at D. Amylose and amylopectin form a complex that is insoluble and so does not affect the water potential of the cell. Again, going back to the question, because this could be correct, right? The hydrolysis reactions result in the breaking of bonds and that releases energy. But the question is asking you how the molecular structure of starch is suited to its function. What is the function of starch? Starch is an energy storage molecule and it is only found in plants. Plants are the only ones that are able to store glucose in the form of starch. And so if it's about storage, then it means that starch at some level has to be insoluble and not affect the water potential of the cell, and that makes your answer D and not C. All right. Okay, let's look at question number 9. Cows and whales are mammals that produce milk to feed their babies. Newborn whales grow faster than newborn cows. So that's important information that I believe we didn't know before. Milk contains fatty acids with a range of chain lengths, and the table shows the percentage of fatty acids of different chain lengths in cow milk and whale milk. It then says which statement about the ratio of fatty acids, 4 to 16, of short fatty acids, 4 to 16 carbon atoms, so these are all short, to long fatty acids in the milk of cows and whales is correct, so these are long. So something I would say here before I even look at the options is for you to remember that the longer a fatty acid chain is, the more likely that it supplies a lot of energy. And so if you think back to if you think back to respiration, in order for you to make acetyl coenzyme A, you usually have 2 carbon atoms that are taken off the chain at a time in order to make that from a fatty acid that then goes into the Krebs cycle and helps with the production of ATP. So in this case, the longer whatever mammal has the longer fatty acid chain, most likely requires more energy. So let's look here. The ratio in cow milk is higher because newborn cows need more energy than newborn whales. This is not accurate. Yes, indeed, the ratio of short fatty acid lengths is higher in cows if you compare that to the amount in whales, but that's not going to give them more energy than whales. The ratio in cow milk is lower. Again, that's not true. The ratio in whale milk is higher. This is the ratio of short fatty acids. That's not true. The ratio in whale milk is lower because newborn whales need more energy than newborn cows. that is definitely correct. So because the ratio of short fatty acids is lower in whales compared to long fatty acids, it means that they require more energy, which is why they have longer fatty acid chains. All right. Okay, let's look at number 10. The structure of phospholipids includes a phosphate, polyphosphate head. Yeah, that's true. Hydrophobic fatty acid chains. That's true. Saturated fatty acid chains. Well, some of the fatty acid chains have double bonds, so maybe not true, but let's still leave that hanging and look at the question. The question says, what are essential for the formation of a phospholipid bilayer in a cell surface membrane? So when we're speaking about something being essential, it means absolutely necessary. And in this case, the only absolutely necessary things that you need in order to make a phospholipid bilayer is to have phosphate heads that are sitting at the top and hydrophobic tails that are sitting at the bottom. And so that gives you A as the possible answer. Then question 13, a student investigated the effect of enzyme concentration on the rate of hydrolysis of protein in milk. When the enzyme and the milk were mixed together. Oh, and I see this is question number 13, sorry. So not number 11, number 13. I've sort of rearranged them in order to conserve space on this video. But all the other questions are in the video, so please don't worry too much about it. I think they were too long to fit on the slide, so I put whatever question could fit on and just put it here. A student investigated the effect of enzyme concentration on the rate of hydrolysis of protein in milk. When the enzyme and the milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. The student investigated 5 different enzyme concentrations and recorded the time taken for the mixture to become clear. What is an appropriate control for this investigation? So think about it. You have an enzyme, all right? So let's try to draw this. We have an enzyme, perhaps that looks like that, and we have a protein in milk that fits into the enzyme. And when you mix them together, then you have a reaction that changes it from cloudy to clear. Something to remember here is that if you're investigating the effect of enzyme concentration, it means that this is constantly changing. All right, you're constantly changing the enzyme concentration, which is what the student did, and then checking how long it takes. What would be a good control for an experiment like this? Well, in this case, you might want to see what happens if there were no enzymes at all, because the enzyme concentration is what you are changing. So if there were no enzymes, then what would happen? And in that case, you can maybe consider option A, where it says carrying out an experiment where the enzyme solution is replaced with water. Option B is not a good answer because it says carrying out the experiment in a thermostatically controlled water bath at 35 degrees. The point here is that it can't be a control because the enzyme would still be there, and this is the optimum concentration. The optimum temperature for an enzyme to react. So that's not a good control experiment. Repeating each experiment 3 times, that's not how to do a control. Using the same volume of enzyme solution for the 5 different experiments, that's also not a good control, because again, you are trying to investigate different enzyme concentrations. So in order for you to investigate different enzyme concentrations, you have to have a solution that is 0 concentration of enzyme. And then you can maybe say, I'm testing 5 milligrams per liter of enzyme and then 10 milligrams per liter of enzyme. But if you don't have that control as a baseline, then it just doesn't add together. I hope that makes sense. Okay, now this is number 12. Again, for some reason, I think I misarranged them, but just try to follow. If you have this paper in front of you and you're looking through, just try to follow it and see what I've done. So question 12 here says the diagram shows a hemoglobin molecule. If you remember from the videos that I've done, the hemoglobin molecule is made of 2 alpha globins and 2 beta globins. So I guess that's what I would ask you to bear in mind for now while we look at the question. Which row identifies different parts of the molecule? Oh, that's a very good one. So here you have the globin chain, and you can see over here, this is 1 here. That's 1. I'm going to make this picture a bit larger. And this is 2, this is 3, and that's 4. So I guess what you're being asked to check here is the different types of protein structures that you're looking at. So let's see, for number 1 over here, that would be, let's see, is this an alpha helix? That's not an alpha helix that it's pointing at, so A is most likely not going to be correct. That's not an alpha helix. Is that the heme group? No, it's not the heme group either. Is it hydrophobic amino acids? The hydrophobic amino acids for hemoglobin are all on the inside, and because 1 is not necessarily pointing to something on the inside, then I would go ahead and say D is most likely not accurate, and so 1 has to be the oxygen binding site, but we can still check ahead to continue to see what we are looking at. So 2, it says 2 is possibly hydrophilic amino acids. That would be correct, because in hemoglobin the structure is a globular structure, and it's globular because all the hydrophilic water-loving proteins are on the outside, and all the hydrophobic water-hating proteins are on the inside. So 2 could possibly be a hydrophilic amino acid. 3, let's see here, 3, it says is the alpha helix, and that's true. That is what the alpha helix looks like. And 4, it then says is hydrophobic amino acids, and those are on the inside of the protein, and you can see that they're quite sheltered from the outer shell, and so B is most likely the answer. Now, some of you might question the technique I've just used and said, well, how did you know to do that? When you have a question like this and you don't want to waste a lot of time thinking about each option, I always say look at what option number 1 is and try to eliminate it. Number 1, for example, here it says A, number 1 is the alpha helix. Looking at A, looking at number 1, you know that that's not the alpha helix. This here is the alpha helix. This is an alpha helix rather. So you know that those are not alpha helices. These are beta pleated sheets, for example. So you know that if number 1 for A is wrong, then A cannot possibly be your answer. And you can do the exact same thing for C and D, and whatever you're left with. So say, for example, you're even left with 2, you can then try to see which one of them fit, all right, in order for you to make sense of it, all right. The reason, for example, if we had tried to do that and we had left, say we had assumed that C is also a possible answer, like, okay, what if 1 is truly the heme group? And then we continued, we can see hydrophobic amino acids. That seems, for number 2, that's already wrong, because if you look at it, they are on the outside, and we know that hemoglobin has all its water-loving proteins on the outside. So that already makes this wrong already, even without having to go through the rest of it. So that is a good technique to use when you have a question like this one. Okay, and now we come to number 11. So I did some creative arrangements just so I could save space on the slides. But let's look at number 11 and see what we are being asked to do here. The diagram shows the molecular structure of a peptide. So remember that a peptide is more or less is what you have when you have combinations of amino acids to form a polymer. So a peptide is the polymer structure, or the polymer form of a protein. So that's our peptide here. And it says which molecules would result from the complete hydrolysis of the peptide. So if we were to add water to this peptide and break down the structure, what are the proteins or the amino acids that we would get in our solution? So what I always say here is, first of all, start with what the basic structure of an amino acid is. And the basic structure of an amino acid is that, okay, it has a carboxylic group at the one end. It has, sorry, I'm about to make an error there. It has an amine group at the other end, right? And if you haven't learned this, please check out my video on proteins. And it has a hydrogen group, all right? When these form a bond, what you will see is that this will be released as part of water, and so this will then be rewritten as that, right? And then you will have an amine group because it's joining to the amine group of the next protein which would then have a hydrogen attached to it, and then it goes on like that, right? So that's what it would look like. And this would be the side chain, and that would be the hydrogen group. So now let's try to look at this amino acid to figure out where exactly the bonds are being formed. All right, so if I'm looking at this correctly, I would say, so I'm just going to erase this circle I've put here, and I'll use a green marker. If I'm looking at this properly, this looks like a carboxylic acid group, so that's the end of the peptide. This here looks like the side chain, that would be the hydrogen group, and this then is one peptide. All right, and you can see that because that one peptide, if you look at this drawing I made here of the amino acid structure, is being joined to the next one, which has a carbonyl group on it, so it's a C double bonded to oxygen. So that means this is the start of another amino acid rather. This would be the side chain of that amino acid. This would be the hydrogen attached to the central carbon, and this again is, I'm going to use a blue marker this time, is a second peptide. Okay, and obviously that leaves us with just this as the last peptide. I should have used a different color there so that it's easier to differentiate. I think I'll use purple. This here is then a different peptide. All right, so that would be the side chain. This is the entire side chain over here. That's the hydrogen, that's the carbonyl group that's forming a bond with the next amino acid. I keep saying peptide, but the whole thing is a peptide. The individual structures are amino acids. Please just have that at the back of your mind and don't worry too much about my mistakes. So now let's look at these. Which ones fit with what we have circled as the possible answers? So let's see for question A. We can see over here CH2, CH2. We don't seem to have this at all amongst the list of peptides, so that's obviously not the answer. I'm switching back to my red pen because marking with purple seems a bit weird. This is not one of the peptides. We don't have a peptide that has two CH2s attached to it. We only have one CH2. So there's no way that that's part of it. So you don't even need to look at all the others again, employing the method of elimination very quickly to remove things without wasting too much time on them. So that's not an answer. Let's look at B. If we look at B, we have, yes, we have that. We have the CH2, we have the NH2, we have the C double bonded to O, and we have the carboxyl group. So yeah, so that seems like a possibility. Let's look at the second one. For the second one, something seems to have gone wrong. We're supposed to have the amine group, and then for the side chain, we're supposed to have CH2 and OH, but what we have here is CHOH and a CH3 attached. So that tells us that B is definitely not the answer. Let's look again at C. So C has our group from the first one intact. For the second one, it has a CH2OH, and for the third one, it has CH3 and it has H, and it has the two ends correct as well. So always make sure you also check for the two ends to be correct as full amino acids, not as amino acids in a reaction, so that you can get the right answer. So C looks like it is the correct answer, but we're just going to have a look at D so that we can put our minds at ease, right? So if we look at D there, again, everything seems fine on the first amino acid. On the second amino acid, things seem okay as well. On the third amino acid, however, it's telling us that the side chain is H when the side chain is actually CH3. So that tells us D is not the answer, and the answer here is C. All right. Let's look at number 14. What determines the specificity of an enzyme? So what makes an enzyme specific, first of all, that you have to bear in mind is its binding site. So what this question is really asking you is what makes up the binding site? What are the bonds that make up the binding site, or the certain conditions that might make up the binding site? So number one, the covalent and other bonding between our groups of the polypeptide. Yes, that's true. Something to bear in mind is that all enzymes are proteins, but not all proteins are enzymes. So have that at the back of your mind. The optimum pH of the enzyme, that doesn't determine the specificity, but it can affect the specificity. So those are two different things. The covalent peptide bonds between the amino acids of the peptide. So the peptide bonds between the amino acids of the peptides of the polypeptide may not necessarily determine the binding site, right? It might just be about keeping the rest of the enzyme in shape. So that's not necessarily true. The substrate molecule doesn't determine the specificity. So in this case, for number 14, I would say that the answer is C. Let's look at number 15. The graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes fruit juice stains. And you can see that there's temperature, and as temperature increases, so does the rate of reaction of the enzyme, and after a certain temperature the reaction drops. I'm pretty sure you've all learned this. Which statements explain the shape of the graph at temperatures higher than T? So at temperatures higher than T, first thing you have to bear in mind is that when the temperature gets too high, then the enzyme will certainly start to have some of its bonds being broken. Okay, and that's what we call the denaturation of the enzyme. So the enzyme starts to denature and starts to lose its shape as bonds are broken. So one would definitely be a good possibility. Number two, there are more collisions between the enzyme and the substrate. That's not necessarily true, because the enzyme is being denatured while this is happening. So there is no way for it to collide successfully with the substrate. So that makes that impossible. The tertiary structure of the enzyme is altered. That's also true. The shapes of the active site and the substrate are no longer complementary. That is also true. So that leads us to our answer being C for number 15. Let's look at number 16. Which statement describes carrier proteins in cell surface membranes? Carrier proteins in cell surface membranes, they are found on the outer surface of the membranes, allowing for cell recognition. That's not necessarily true. You have what you call your glycoproteins that are for cell signaling, and lipoproteins as well that are for like recognition and cell signaling. So A would not be correct for carrier proteins. Carrier proteins are involved in moving substances through the membranes by either active or passive transport. That is correct. Carrier proteins help to move certain substances through the cell membrane in a way that prevents those substances from interfering with the cell membrane, or allowing them to pass through if they are normally not able to. Carrier proteins allow for the binding of ligand molecules. That's not true. Again, the ligand molecules will most likely be on like a glycolipid or glycoprotein, something along those lines. Carrier proteins are involved in moving substances through the membranes by passive transport through water-filled pores. Those sound more like channel proteins. So carrier proteins can move substances either by active or passive transport. So that makes the answer for number 16B. Number 17, and I know this is a bit long, but I hope that you are following and you are taking note of the explanations as we go along. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential than the water potential inside the cell? So if the water potential outside of the bacteria is higher, remember that bacteria don't have a cell wall that can prevent them from taking in water, or even just prevent net movement of water, or prevent them from bursting. So in this case, if the water potential is higher, so this is our poor little bacterium, and we've put it in a solution of pure water, the water will start to move into the bacterium because the water potential is higher. So I'm just going to say higher water potential, and over here water potential is lower. All right, so remember, osmosis is the net movement of water from a region of higher concentration to a region of lower concentration. All right. Let's look at number 18. By which processes do hydrogenate carbon ions leave red blood cells? Hydrogen carbonate ions, sorry, not hydrogenate carbon ions, hydrogen carbonate ions. I am quite tired. I need to actually make sure I finish this video before I fall asleep and start to snore while the rest of you are listening. So number 18, by which process do hydrogen carbonate ions leave red blood cells? They don't leave through active transport, that's for sure. They don't leave by endocytosis. Again, if something can't leave by endocytosis means that it is being engulfed. They don't leave by phagocytosis either, because phagocytosis has more to do with the intake of bacterium and things that invade the cell. So they leave through facilitated diffusion. And I think this is something that you learn in chapter 8. So if you're not really sure, please make sure you check out chapter 8 to be able to understand that a little bit more. In an experiment, fluorescent dyes were attached to proteins on the outer surface of cell surface membranes. Fluorescent dyes of one color were attached to proteins of a living human cell, and fluorescent dyes of a different color were attached to proteins of a living mouse cell. The human cell and the mouse cell were then fused to form a hybrid cell. This would be what you call a hybridoma, I'm assuming. If you have watched the videos on immunity, you might have come across that. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. What does this experiment show? What this experiment is showing you is that proteins can move around, because it says here they were distributed randomly across the hybrid cell surface membrane. So it's not saying they were inside the cells, but across the cell membrane. Now, a good technique with which you can answer questions, something that has just popped into my head now, is that when you're answering questions, particularly in paper 1, always remember that the questions are typically often arranged in order of the chapters that you're studying. So for paper 1, you write on chapters 1 to 11. And the questions will typically follow that order from chapter 1, 2, 3, 4. And you know that chapter 4 is cell membranes and transport. So that might give you an idea of how to answer this question. But even if you were not thinking about that, you can also just pay attention to the words that are being used here. So it says here, what does this experiment show? Proteins are found only on the outer surface of cell membranes. That's not necessarily true. Proteins in the outer layer of a bilayer do not penetrate to the inner layer. That's not necessarily true either. Proteins move freely in the phospholipids of a bilayer. That is true. And the cell surface membranes of the two cells are bilayers. Well, that could also be true, but that's not what the experiment is showing us. So the answer here would be C, because it basically says that the proteins were distributed randomly across the hybrid cell surface membrane. And if you remember about the fluid mosaic structure of the cell membrane, we speak about how the proteins are scattered if you're looking at them from the top in order to make it look like a mosaic. So that's how you would think of this question. Okay, number 20 shows the mitotic cell cycle, during which phase is DNA replicated. So the replication of DNA in the cell cycle typically would happen after interphase, okay? And interphase is the longest phase. So it would happen somewhere here. All right, so that would, I think, be B. So the answer should be B. So you have interphase, you have your G1, S1, G2 phase, and then G1 phase, S phase, G2, and then you have your cell division, and then you have other things that happen afterwards. So the answer here would be B. Let's look at number 21. Bacterial cells divide by a process called binary fission. What binary fission means, for those who might not know, is basically bacteria don't go through a long division process. They just split into two, which is why you can make many bacterial cells in a short period of time, because it's just about giving them enough food and they split, and they are happy to split into two. So which macromolecules must be synthesized for binary fission to happen. In this case, you obviously need your cell membrane proteins and the RNA, otherwise the splitting that happens would not yield a viable cell. You also need it to have a cell wall, which is made of peptidoglycan, and to have the DNA so that it can be a living cell and also be able to replicate itself. You don't need it to have enzymes, and bacteria certainly do not have cellulose, so you can't say 3 is an option, so that leaves you with B as your answer. Let's look at 25. Again, I've skipped here, so we'll be missing 22, 23, and 24. That's probably because they were too long to fit on, and I was trying to minimize space as much as possible. Which statements about mRNA are correct? So question number 1, mRNA can form base pairs. That's true. mRNA can form hydrogen bonds. That's true. mRNA is single-stranded. That's also true. So I would just go ahead and pick the answer as A, because all of them are true. All right, and when it says base pairs, remember that with mRNA you have A binds to U and you have C binds to G. Okay, and the only way this is different in DNA is that A binds to T in DNA. Okay, now let's go back, rewind, and look at number 22. The electron micrograph shows a group of human chromosomes. Which label is correct for each of the structures labeled WXY? All right, so let's see. W is pointing right to the center of the chromosome. So that's not the centriole. The centriole is actually not found in the chromosome. It is found in the cell. So just because W is wrong for A and B, I'm going to go ahead and cancel out A and B as possible options, and this helps to narrow it down. Again, when you get into the exam, this is a technique that you should use because it will be very, very helpful for you. So I'm just going to go in there and look at what happens with C and D. So I know that W is definitely the centromere. All right, so I'm just going to take that. I know that X is also referring to the telomere, so I'm going to pick that as well. And I know that Y is referring to the chromatid and not the microtubule. So in this case, my answer for number 22 is C. It's not the microtubule. All right. Which statements, this is 23, about the behavior of chromosomes during mitosis is correct? It says they also attach to spindle fibers to contain them within the nucleus. They condense to prevent further translation of genes. They reach the poles of the cell and begin to uncoil. They replicate to produce sufficient DNA to form 2 new nuclei. The behavior of chromosomes during mitosis has more to do with this C, all right, and not with D, because in D, by the time they're replicating, that's not part of mitosis. That's actually another part of the cell cycle, which we just spoke about, the G1 S phase and the G2 phase. If you want to understand that in detail, please watch the video I did on mitosis. They attach to the spindle fibers to contain them within the nucleus. That's not true. They condense to prevent further translation of genes. That's also not true. So the answer has to be C over here. And let's look at 24. Remember, we already did 25. So we're sort of moving around very randomly here, but I guess you are following, and I hope it's helpful. Gene mutations can occur by substitution, deletion, or insertion. What is the smallest part of a DNA molecule that can be changed by a gene mutation? So in this case, the smallest part that you can change of a DNA molecule is actually the nucleotide. That's what you're able to change. If you have an insertion or a deletion or whatever, it's going to change the nucleotide, and if the nucleotide is affected, then you basically end up changing the codon, the base, and then you change the gene. All right, and if you want to know the differences between the nucleotide, the base, the codon, and the gene, just post the question in the comments. And again, as members, you have priority responses for comments, so I will have to respond to you. Okay, now we're making progress, and we can see we've moved away from chapter 5, which was mitosis, and we're now in chapter 6, which is DNA replication and protein synthesis. And it says here, the table shows the role of 4 different proteins involved in DNA replication. And you have the proteins, which are helicase, topoisomerase, single-strand binding protein, and the DNA polymerase, and it tells you the roles of these proteins. So the helicase unwinds the DNA helix, the topoisomerase breaks and joins the DNA strands, the single strand binding protein binds to separated DNA strands to stabilize them, and then you have DNA polymerase, which synthesizes the DNA strand. This is quite an odd question because it's already told you the role, but then it goes on to ask you which row shows the function of these proteins. So let's again use the method that I've been speaking about, where I say first look at the very first one and see which options are incorrect for that one. So for helicase, we know helicase is the one that unwinds the DNA double helix. So here adds DNA nucleotides to the 3' end of a growing polynucleotide. So we know that A can't be correct because that is not what helicase does. Enables tension caused by unwinding to be released. That's not what helicase does. As a matter of fact, helicase is the one responsible for unwinding. And then C enables tension caused by unwinding to be released again. So that already, can't be correct. So we are left with just one, and let's see if that one ticks all the boxes. So helicase makes strands available as templates. Yes, that's true, because when it unwinds them, it opens them up so that DNA polymerase can bind. Topoisomerase is the enzyme that sort of releases the tension that is caused. So when you're unwinding on one side, you tend to make it a bit coilier on the other side, and so topoisomerase helps to relieve that tension. So that is correct. The single strand binding protein prevents original strands from reforming complementary base pairs. Yes, that's correct. And you have the DNA polymerase that adds DNA nucleotides to the 3' end of a growing polynucleotide strand, and that leads us to D as the answer. So again, you can see that this technique works. Simply focus on one and try to eliminate rather than trying to go through every single one. All right, let's look at 27, 28, and 29. Which type of sugar and which type of bond are found in a DNA molecule? So in a DNA molecule, you have what you call deoxyribose. That's what the sugar is called. It's called deoxyribose. Again, please watch the videos that I've done on these chapters. And deoxyribose is a monosaccharide. It's not a disaccharide, it's not a polysaccharide. So already you can just go ahead and eliminate those ones. All right. And because it is a sugar molecule that is formed, that is bound to a phosphate and all of that, there is no way it would have a peptide bond, because peptide bonds are for proteins. And that just leaves you with the answer D. See how easy that is? Let's look at 28. 28 is the one on the right here. So now we're moving into transporting plants. And for this question, I just have to say, please study what these look like. And always just remember where the root is, what the stem is, where the xylem is, and where the phloem is. All right, so that you can have it correctly. So it says here the diagrams represent transverse sections of 3 plant organs, the root, the stem, and the leaf. Which row is correct for the phloem? So if you're looking for the phloem here, you should always remember that the phloem is the one that is typically on the outside in the root. Over there, well, this is the xylem. The xylem is sort of like a hollow structure in the center. So you know that 2 is correct for the first one. So you can go ahead and eliminate A and B. All right, and again, if you look at the stem, again, it's going to be 3 because it's on the outside. And if you're looking at the leaf, it is going to be 6, all right, because it is, this one's not inside or outside. This one I sort of memorized when I was teaching the subject, so I'll just go ahead there and say it's that. Anyway, they've given you a clue here because they've said here that the stem is number 4, which is obviously wrong. All right. Now let's look at number 29. Sucrose moves from a phloem tissue, phloem sieve tube element into a root cell, which changes to the water potential and the volume of liquid in the phloem sieve tube element are correct. So when sucrose moves into the root cell, what's going to happen in that case is that you will have less concentration of solutes inside the phloem, which means that the concentration of the water becomes higher because you're removing the sugar. Okay, and so if the concentration of water is higher, that means it will have a higher water potential. However, that doesn't mean that the volume of the fluid increases, because when you have fluid moving out, it actually sort of decreases the water. So that then tells you that the answer here should be A. And obviously the water potential doesn't become lower, so that's not going to be true. The reason why the volume of the liquid decreases in the phloem sieve tube element is that when the water moves, when the sucrose moves out and moves into the root cell, there's going to be more water sort of moving into the root cell, because again, it's trying to, this now becomes an issue of diffusion, right? And so this increases the water potential of the phloem sieve tube element, and that allows water to move across. If that just confused you, please just check out the videos that I did on transporting plants. I promise you the explanation there is a lot better and a lot more straightforward, and much better than tired Dr. Demi. Okay, number 30. We've done about 3/4 of the paper, so we just have a few left to go. Let's do these ones. Which statements about water movement in plants are correct? Water can pass through cell walls containing layers of cellulose fibers. That is true. Cellulose fibers do allow water to pass through. They are not regulatory. The cell wall doesn't regulate what goes in and out of a plant cell. What it does, however, is that it prevents the plant cell from bursting if it takes up too much water. Okay. And then number 2, water can pass through cell walls containing rings or spirals of lignin. Yes, that's true. And it can't pass through a band of suberin in cell walls. That's also true. I will try to do a short video on suberin. If I forget, please just post a comment here and I will get to it. So the answer for number 30 would be A. And then for number 31, the photomicrograph is a transverse section of a leaf. This is something I often tell students to just memorize in terms of the images of xerophytes and what they look like, because you might know the different things that make up a xerophyte, but you might not be really sure of what they look like in a photomicrograph. However, let's say that you don't have the bandwidth to memorize it because you're just tired, you've been studying for a long time. So, I mean, what can you do? I would say if you're being asked to label or to find out structures that are related to the function of the xerophyte or the adaptation of the xerophyte, look around the surface of the xerophyte. So one part to look at here, for example, is that this part is sunken. Okay, so you know that this is like part of an adaptation. So you're going to circle 1. This thin extended structures, they're also on the outside. That's 2. These on the inside are not necessarily going to do anything in terms of its adaptation, but here on the outside you also have what perhaps might be a thin cuticle or whatever it might stand for, but the point is that it's on the surface. Most of the adaptations of the xerophytes that you're usually asked to address are on the surface, and so you will have to pick whatever is out on the surface. And so for this question, I will confidently pick C, because I've seen questions like this before and I know that they are usually on the surface. So if you don't know what it looks like, you can't remember, look around the surface of the photomicrograph and pick the structures that relate to that and try to find that in the options. And something one of my colleagues used to say to his students that used to make me laugh but actually makes absolute sense. He would say, if you're not really sure, think of what you know about it and then try to guess the answer based on that. So that's good advice. I mean, do with it what you will. So here, a maize crop is growing successfully in a field in which the water potential of the soil is minus 40 kilopascals. What is the most likely water potential of the cell sap in the root hair? So in this case, well, typically you're not often asked to calculate this kind of water potential. So this is quite a surprise, but let's think about it. So the water potential of the soil is minus 40 kilopascals, which means that the soil has less water in it. Okay, the soil has less water in it than the plant would, because the water potential is quite low. Or well, that's what we can assume. What is the most likely water potential of the cell sap in the root hair? So the cell sap probably is not going to be 0. Because 0 water potential already suggests that there is some balance on both sides, and that doesn't seem to be the case. The water potential is also not going to be minus 20, because that suggests that the water in the root hair is higher than the water in the soil, which is impossible because it's the water from the soil that moves into the root hair. So if the water in the soil is lower, the root hair would also have low water potential. It can't be minus 40 because they're probably not at the same water potential. So I would say your best guess in this case would be minus 60. It's just to show that the root hair would have a lower water potential than whatever is in the soil, because if the soil has lower water potential, then there isn't going to be a lot moving into the root hair. So yeah, that's something to just bear in mind there. Let's look at 33 and 34. The contraction of the heart is coordinated through electrical impulses passing through the cardiac muscle. What is the right order or the correct order of the sequence of these impulses? This is one of my favorites. And again, probably not the right place to go through the entire heart in detail, but you should remember that the sinoatrial node is the one that helps to set off the impulses and to sort of control how the excitations happen. So you're going to just maybe cancel out A and D, and you're left with B and C, and it goes through the right and the left atria. Yes, that is where the blood goes through first, and then there's some squeezing that happens that goes through the atrioventricular node and then goes to the porcine tissue, and that is how the excitation wave travels. So the porcine tissue is the last bit and not the atrioventricular node. The atrioventricular node is actually activated after the atria contracts, so that would then make it B, because after the atria contracts, then it's the ventricles, and then it goes actually it's from the ventricles into the atria, and then from the atria it is squeezed out to other parts of the body. So this is not the full sequence node, the full set of the full sequence, but it does give you an idea. So just be sure to know what the full sequence is, and so even if some bits are missing, you're still able to fill in the gaps there. All right, which features allow the aorta to withstand high blood pressure? I think this is one of the questions my students love the most. It has collagen fibers and it is also elastic. All right, so that's something that helps it. Collagen fibers and smooth muscle. The smooth muscle doesn't let it withstand high blood pressure. It lets the blood flow within the aorta without any restrictions. So that's what the smooth muscle is for. It's not the endothelium, so endothelium and smooth muscle are certainly not what you would consider here. All right, which row is correct? So we're looking at 35, 36, and 37. Which row is correct for the pulmonary artery? So always remember, whatever is an artery carries blood away from the heart, and if it's carrying blood away, then it is dealing with something that is either oxygenated or deoxygenated, depending on where it is going. If it's going to the pulmonary, so let me explain it this way. If it's the pulmonary artery or the pulmonary vein, it is connected to the lungs. The arteries always carry blood away from the heart. So the pulmonary artery is most likely carrying blood away from the heart towards the lungs. So if it's carrying blood away from the heart towards the lungs, it means that the blood it is carrying is deoxygenated blood, and it is carrying that blood to the lungs so that the blood can become oxygenated. So that means that A and B cannot be correct here. It has to be either C or D. All right, so just bear that in mind. Because it is an artery, it's going to have a thick wall because it carries blood at a higher pressure, and the lumen size is also going to be small because that helps with the pressure. The lumen cannot be large. If it was thin, the vessel might be easily collapsible, and that then makes it unsuitable to carry out its function. So the answer here would be D. Let's look at 36. The graph shows the oxygen dissociation curves of adult hemoglobin in different carbon dioxide concentrations 1 and 2. Which conditions could change the shape of curve 1 to the shape of curve 2? So this is the shape of curve 1. I'm not going to go over this in detail because I think I did a good explanation of it when I explained this in chapter 8. So make sure you check out that playlist. But this is basically talking about what happens, the percentage saturation of hemoglobin when you start to increase the concentration of oxygen. And as you can see, as you increase the concentration, hemoglobin becomes increasingly saturated in scenario 1. However, when you're dealing with scenario 2, you can see that hemoglobin is not as quickly saturated. So that means that something is tampering with that. And only two things can tamper with that. It's either carbon monoxide or carbon dioxide. When you have carbon monoxide, then the graph is actually a lot worse than this, because carbon monoxide is a non-competitive inhibitor that terribly affects how hemoglobin binds with oxygen. But in this case, carbon dioxide, so you can't say it's decreased carbon dioxide, because it's certainly not that, but increased carbon dioxide concentration causing a decrease in pH is a most likely answer. And the reason for that is that the way the body removes carbon dioxide through hemoglobin is that it binds with water and it forms an acid, and hemoglobin then tries to mop up that acid so that the blood doesn't become too acidic. If that was confusing, just check out the video on chapter 8, but the answer there would be A. And number 37, what maintains the necessary concentration gradients for carbon dioxide and oxygen in the lungs? Obviously, it has to be the good ventilation of the lungs. That's what helps with the gas exchange. So whenever you breathe in and you're breathing out, you're taking in oxygen, you're breathing out carbon dioxide. That's helping with the crossing of these gases. I would also be tempted to say B looks like a very correct answer, that the large surface area of the alveoli, but having a large surface area doesn't necessarily mean that you are maintaining a concentration gradient. So a concentration gradient requires you to have a high amount of one thing and perhaps a low amount of the other one. A high amount of one thing on one side rather, and a low amount of the same thing on the other side. So the surface area doesn't necessarily help you address that. So I wouldn't pick B as my answer. I also wouldn't pick C, and I wouldn't pick D, because D just more or less allows for easy diffusion, doesn't necessarily maintain a concentration gradient. So I would go with A as the answer here. All right, we're close to the end. Which cells and tissues are present in the trachea? So in the trachea, you know for sure that you have the cartilage. The cartilage is present. You also have the ciliated epithelium, and you have goblet cells that produce mucus. So I don't know why this is even a question. It's so easy. The answer there would be B, because B is the one that encompasses all of those. And number 39, which is more or less our second to the last answer, last question rather. The global mortality figures for some diseases in 2013 are shown in the table, and this is how many millions of deaths. And it says how many millions of people died in 2013 from the bacterial diseases listed in the table. So this question is not that tricky, but what they are asking you to do is not just to look at the numbers, but to first of all determine which ones are bacterial, which ones are viral, and which ones are caused by something else like a product test. So you know that HIV/AIDS is caused by a virus. At least I hope everyone knows that by now. Tuberculosis is caused by a bacterium called Mycobacterium tuberculosis, and malaria is caused by a protoctist called Plasmodium, and cholera is also caused by a bacterium called Vibrio cholerae or cholerae. So this is also another bacterium. I'm just going to write BACT over there. So all you have to do in this case is then add the value for TB and the value for cholera, and you will get a B there, or the answer should be B at least. The last question, and I've deliberately made this big to signify that we've made it to the end. Why is it difficult for B lymphocytes and T lymphocytes to respond to the antigens on pathogens that are intracellular parasites? And I'm guessing they're asking this because of COVID maybe. But by the way, speaking of which, you should expect some COVID related questions, especially towards the end of this paper and possibly in your paper too, because I imagine that they would want to use some of the data that they've gotten to test how you are able to analyze viruses and so on. So please bear that in mind. All right, and don't listen to conspiracy theories to answer the questions. Make sure you're listening to the actual thing. So let's see over here. The antigens are constantly mutating. That's not true. Antigens don't mutate. So always remember that the antigens are what's present on the surface of the cell. All right, so if this was some scary cell, those lines on top of it would be antigens, and they don't necessarily mutate. The antigens can destroy the lymphocytes. That again is not necessarily true. The lymphocytes do not encounter the antigens. Yes, that's correct, because it says they are intracellular parasites. So it means they are hidden within cells, and for that reason, the lymphocytes are unable to see them, and so they are unable to defend the body. If they were to see them and recognize them, then certainly they would be able to destroy them. So that brings us to the end of this video, and I hope that you have enjoyed it. Just some closing remarks. Your paper 1 has changed in 2022. You now have an hour 15 minutes, which is what we've done. We've done it in less than an hour 15 minutes here on the video, but I encourage students to try to do it within an hour so that they can spend the last 15 minutes really checking their answers. The second thing I want to say is that with paper 1, students tend to be really overconfident, and for that reason tend to make a lot of mistakes. As a matter of fact, even my best students, and these include my students who got straight A's, made most of their mistakes in paper 1. So paper 1 is not as easy as you might think. Sometimes the options look really similar, and I know we've done the specimen paper, but usually also bear in mind that the actual paper that you get is sometimes harder than the specimen paper. So please don't stop practicing. Have a look at past questions. I'll do a couple more past questions on this, but just thought to put this out to help you and to encourage you, and also to say thank you for signing up for memberships. Obviously, as more people sign up, I will be doing a class on that. I'll be doing a live stream class so that we can have a mini classroom structure and just see what we can get done together and what we can learn together. Again, thank you all so much for subscribing and for supporting the channel. I really appreciate it, and I wish you good luck in your exam preparations. Goodbye until the next time.